Hi everyone, my name is Jacob Nearing. I'm a PhD candidate at Dalhousie University under the supervision of Dr. Morgan Langell. And this year I have the great pleasure of being one of the teaching assistants for the CBW 2021 microbiome workshop. In this lecture, we will be talking about statistics and some machine learning in the context of microbiome research. The goals for this lecture are: one, to introduce common tools for answering questions in microbiome research. These tools include statistical frameworks such as PermaNova, ANCOM, LDEX2, and linear models. We'll also discuss the idea of compositional data analysis versus the more traditional proportional or relative abundance-based analysis. And finally, we'll briefly introduce machine learning in the context of microbiome research. I would also like to highlight the goal of this lecture is not to get down into the nitty-gritty of each statistical tool, but rather to give you an idea of what tools are out there and what you can do with your data using these various tools. And so, in the previous lectures, you've learned to generate a number of different metrics from raw 16S ribosomal RNA gene sequencing data. This includes alpha diversity metrics such as richness or Shannon diversity. It also includes various beta diversity metrics such as unifrac or Bray-Curtis dissimilarity. Finally, you also learned how to generate read counts of various ASVs or amplicon sequence variants and how to classify those variants as different taxonomy or microbial organisms. Numerous discoveries have been made through these various metrics that you've just learned to generate. And one of the most interesting ones that I've come across in the last 3 years is the relationship between the human gut microbiome and cancer immunotherapy. So in the year 2018, 3 papers were published relatively close to one another that uncovered this idea that the gut microbiome may be associated with how well cancer immunotherapy works in humans. So in this one paper in 2018 by Rodi et al., they found that when they took fecal samples from individuals that either responded to immunotherapy to melanoma or didn't respond, they found that responders had higher abundances of the bacteria Akkermansia mecinophilia than non-responders. They also found that non-responders had higher levels of this Parabacteroides species here. In a similar paper that was published quite within the same time frame, they showed that if you were to take stool samples from either responding patients or non-responding patients and give them to germ-free mice, so you would colonize these germ-free mice with the microbes that were found in the gut of these melanoma patients, and then give these mice melanoma cells to model cancer, and then treat these mice with immunotherapy, in this case, anti-PD-L1 therapy, what you would see is that mice that received stool from responders actually lived much longer and had smaller tumors than mice that received stool from non-responders, indicating that, again, the gut microbiome must or could play some role in the efficacy of cancer immunotherapies. And the reason why I bring this up is that this research wouldn't be possible without the use of various statistical tools that can be used to identify interesting microbial patterns. However, before we get into introducing these tools, I'd first like to talk about this idea of compositionality and how it relates to the microbiome. So when you sequence a sample, what you'll notice is that samples will have varying amounts of reads in them, or a varying read depth. And unfortunately, the read depth within a sample is not a direct representation of the amount of DNA found within the sample, or even the amount of cells found within that sample. And so what this inherently means is that one read does not equal one cell in your sample. And if you have a sample that has 20,000 more reads than another sample, it doesn't necessarily mean but the sample with 20,000 reads has 20,000 more cells or copies of the 16S gene than the other. And so this creates a problem when we're examining microbiome data, because you could imagine if we're comparing all these samples and they have varying read depths, and these read depths are not necessarily proportional to the actual cell counts in the sample, we could make some false inferences. So one way to solve this is actually to treat our data as proportional values or relative abundances. And so what I mean by this is, instead of looking at the raw number of reads that one genera may make up in a sample, you look at the percentage of reads that genera makes up. So based on this analysis and looking at microbiome literature, you'll become quite common with these stacked bar charts. So this is a stacked bar chart I made last year, looking at the composition of the oral microbiome of Atlantic Canadians. And basically on the y-axis, we have the relative abundances, which go from 0 to 1. And on the x-axis, we have different samples. And so what we can see in this plot is, on the right-hand side in green, you can see that there's a lot more Villanellia species on the samples on the right. And on the left, there seems to be a higher proportion of Neisseria species that make up the oral microbiome on these samples on the left. And so this is a very common way of viewing microbiome data. So instead of looking at raw counts, we're looking at the proportion that a specific taxa or specific amplicon sequence variant makes up of the total number of reads found in a sample. And so while that solves this issue of one read not equaling one cell, it still doesn't solve the issue of uneven read counts. And the reason for this is because we run into this issue of an unequal sampling effort. And what I mean by this is, you could imagine if you were an ecologist and you were out in a field, and on the first day, you spend an hour in that field surveying the different animals that come across and drink from a watering hole. And say on the second day, you do the same analysis, but instead of spending 1 hour, you spend 5 hours. Well, you could very well imagine that on the second day, you would find a higher diversity of the number of different animals that visit that watering hole. However, this isn't because on the second day, there was some interesting event that occurred, but rather because you spent 5 times the amount of time surveying the land. And so this can be the amount of time that you spent surveying the land can be akin very much to the sequencing depth of the sample. So if you were, for instance, to sequence a sample with a depth of 10,000 reads versus 1,000 reads, you would expect to find more unique microbes in 10,000 reads than 1,000. And this is exactly what you see in these plots that we call rarefaction plots. So on the y-axis, we have the number of unique observed amplicon sequence variants, and on the x-axis, we have sequencing depth. And as you can see, as sequencing depth increases, going to the right, we see that there's an increase in the number of uniquely observed amplicon sequence variants. And so this creates a problem if we're comparing a sample with, say, 4,000 reads to another sample that has 20,000 reads. So one way of solving this issue is through this idea called rarefaction. And rarefaction is the process of basically taking the samples and randomly subsetting them to all have the same read depth. So in this case, perhaps we're interested in subsetting all of our samples to 6,000 reads. And generally, the number that this is chosen is based not only on when this rarefaction plot begins to plateau, but also based on the minimum, based on your samples and which samples have a reasonable number of reads that you'd want to include in your analysis. And so rarefaction generally should be used when generating alpha diversity metrics, such as Shannon diversity, richness, and evenness. And it should also be used when generating many of the common beta diversity metrics we use, such as unifrac distances or breakers to similarities. However, there is still something else we need to take into account when looking at proportional analysis. And this is the fact that the abundance of one microbe, relative abundance of one microbe is dependent on the relative abundance of all other microbes. And what I mean by this is if we were to take a community, this community 1, and we have 3 microbes in it, Staphylococcus, Bacteroides, and Akkermansia, and they all have the exact same absolute abundances. So if we plotted the relative abundance, we'd see they all make up 33% of the sample. Now, say we take a second community and we increase the number of Bacteroides cells by 4 times the amount. So now we have 4 Bacteroides cells, 1 Staphylococcus cell, and 1 Akkermansia cell. If we plot the relative abundances, you'll see that indeed Bacteroides does increase in relative abundance. However, something that is important to recognize is we also see the relative abundance of Staphylococcus and Akkermansia decrease. This is despite the fact that their absolute abundances between community 1 and community 2 did not change. And so when doing proportional based analysis, it's important to recognize that you're looking at how they're looking at changes in the relative abundance, and this does not necessarily mean that the same directionality of change occurred in absolute abundance. So in this example, we see that despite Staphylococcus staying the same in community 1 and community 2, it actually decreased in relative abundance in community 2. So that's an important caveat you need to take into account when examining proportional based analysis or relative abundance based analysis. Fortunately, in the last few years, there has been a push to kind of solve this issue through the use of what we call compositional data analysis frameworks, or CODA. And these methods are based on ratios rather than proportions. And there are a number of different ratios that CODA methods use, but the 2 I will highlight in this lecture are center log ratios and additive log ratios. The center log ratio measures the ratio between the abundance of a microbe of interest and the abundance of the average microbe in that sample. And what I mean by average abundance of the average microbe, I mean the geometric mean of all the abundances of each taxon in the sample. The formula you can see here on the page. So this gives us an idea of basically, if our taxon of interest is Akkermansia, we can see whether or not Akkermansia is more common than the average microbe or less common than the average microbe in the sample. So this is a pretty powerful metric that we can use. The second metric is called the additive log ratio. The additive log ratio, rather than using the average microbe as the denominator, you instead pick a reference taxon. And this reference taxon, generally, you would want to be present in all the samples you are examining, and also not to vary in the cell count or the biomass of the samples you are interested in looking at. And this is important because basically, if you know that this taxon is always present in a sample and at the same number of cells, or generally around the same number of cells, you can then compare other microbes, the read counts of other microbes in that sample, to see whether or not there are more of that microbe in compared to your reference taxon, or less of that microbe in compared to your reference taxon. And because your reference taxon doesn't change in abundances, you can then compare these across the samples of interest. And so the formula for this metric is also shown here in the slide. While CODA methods have been extremely useful, there is some downsides, and that is because one of those is that it can be very difficult to interpret what the actual meaning of the results are. For example, in the center log ratio, it's not quite clear what does it mean by the average abundance of a microbe in the sample. What is that abundance exactly? The other thing that is difficult about these methods is when doing an additive log ratio, it's a lot of the time very unclear which taxon you should pick as a reference sample, especially in samples that you haven't been very well characterized. So some environmental samples that not very many people have sequenced before. And so despite this, there are many examples of CODA analysis in microbiome literature. So in beta diversity, we have Atchinson's distance and robust Atchinson's distance, which is just an add-on to the previous Atchinson's distance that makes it a little more accurate for highly sparse microbiome data tables. And then we also have two tools that we're going to, one of which we're going to highlight in our tutorials for differential abundance, and that is LDEX2 and ANCOM. So if you're interested in learning a little bit more about what it means for microbiome data sets to be compositional, I highly suggest reading this paper that came out of Greg Galor's group, which highlights why it's important to take this idea of compositionality when examining microbiome data sets. So with that out of the way, you may be scratching your head wondering, well, there seems to be a lot of caveats. So what exactly should I be doing with the data that I'm generating? And so in the following section, I'm going to give you some practical examples of different statistical tools or tests that you can use to answer some common questions that might pop up during your microbiome analysis. So for example, perhaps we're looking at the alpha diversity of two different groupings of samples, and we're interested in wondering whether or not there's this statistical difference between the richness in one group compared to the other. Well, they're kind of, if we only have two groupings, we can use what's called the Wilcoxon-Rank sum test, or if we have more than two groupings, we could even use what's called the Crisco-Wallace test. And both of these tests are non-parametric tests, so they don't depend on the data being normally distributed, and can test whether the alpha diversity between two groups or even multiple groups in the Crisco-Wallace scenario differ in diversity. The second question you might be wondering is, well, perhaps I did my alpha diversity analysis, but it includes samples that came from multiple different rounds of sequencing or multiple different rounds of DNA extraction. And I want to include that as a confounding factor in my analysis. Well, one way you can answer this is through the use of linear models. And now I don't have a lot of time to talk about linear models, but I'm sure many of you have learned about them. And if not, there are plenty of resources online to do a crash course in linear modeling and learn about how to control for confounders in linear models. But in general, what you're going to want to do is include the confounding variable, in this case, DNA extraction, as a covariate. Finally, perhaps you're interested in examining data over some sort of time scale. So in this example, we have the richness, the number of unique ASVs in the sample. And then on the x-axis, we have the number of weeks that have passed. There are multiple ways of analyzing this, and actually, fortunately, there's actually a plugin in QIIME 2 called the longitudinal plugin, which is strictly made for analyzing these kinds of scenarios, and it actually makes use of linear mixed effects models. And so again, we don't have time to jump into linear mixed effects models, but if you run into a scenario such as this, I highly suggest doing some reading about linear mixed effects models, and I'd also be happy to chat with you either during the workshop or after it about how you might go about implementing that for your project. The next type of diversity you might be interested in looking at is beta diversity. So on the plot here, on the left, we have a principal coordinate analysis plot. And we have two different types of samples here. We either have samples that have faced an infection, which are highlighted in red, or we have samples that haven't faced an infection, which are highlighted in blue. And the question we want to know is whether these two groups differ in beta diversity or overall community makeup, depending on whether they had an infection or not. So one test we can use is called a Permanova test, and it tests whether the distances within a sample grouping are more similar than outside that grouping. i.e., it helps you answer the question, do these two groups significantly differ in beta diversity? And this type of testing is implemented in QIIME 2 through the beta group significance plugin, which I'm sure you'll be testing out during the 16S tutorial, as well as the vegan R package as the function Adonis. With Permanova, it not only tests whether or not the location, so the centering of the data, so where the centering of the data located, but also how well the data is dispersed compared to the other group. So one thing you should always complement your Permanova testing is with a beta dispersion test. And what a beta dispersion test does is it tests whether the dispersion from the centroid is similar between the same groups. So you can imagine the red group has a centroid where my red marker is here, and you'd want to test whether the spread of data from that centering is similar from the spread of data from the centroid in the blue group, which maybe exists somewhere where my red marker is here. So this test is implemented in the vegan R package beta dispersion as the function, beta dispersion. And in combination with Permanova, it helps you determine whether the centroid or the dispersion of the data differs between the two groups of interest. So for example, say you tested your data and you had a significant Permanova and a non-significant dispersion test. This would indicate that the centroid, so the centering of where that data exists, is different rather than the dispersion of that data. Now, one of the most seemingly easy, but actually one of the most difficult questions to answer in microbiome analysis are which taxa or amplicon sequence variants significantly differ in abundance between my groups of interest. So for example, between healthy individuals and diseased individuals. And before we even talk about the tools to answer these questions, it's important to spend a little time thinking about what you're exactly asking here. You need to remember back to the previous slides that there are kind of two different types of abundance that you could be comparing here. You could be comparing relative abundances or a proportional based analysis, or you could be looking at compositional abundances using ratios, such as the center log ratio or the additive log ratio. And there are a number of tools that can be used to answer these questions based on whether or not you're interested in looking at relative abundances or compositional based abundances. In a proportional analysis, you could use tools such as Maslin-2 or MetaGenomeSeq. Unfortunately, we don't have time to talk about how these two tools work, but they have great tutorials and they both have two really good papers outlining how they work. And in a CODA analysis, there are two other tools I mentioned previously that could be used for differential abundance testing. The first being ALDEX2, which actually can use a number of different CODA metrics or ratios, but the most common or default one is the center log ratio. And it's actually unique because it actually uses a subsampling approach based on a Dirtlich multinomial distribution or a probability distribution that lets it estimate how likely taxes to be differentially abundant between the two groups of interest. And this can be done either using Wilcoxon tests or even general linear models, which will allow you to incorporate confounding variables. It should be noted, however, that this subsampling approach does make it a little less sensitive to changes in rare taxa. So taxa of low abundance would generally not be indicated as being significantly different than using ALDEX2. A second tool that we'll actually be using during the 16S tutorial is ANCOM. And ANCOM uses additive log ratios, but instead of picking a single reference taxon, it actually chooses every single taxon to be used as a reference taxon. I.e., it takes say Akkermansia as the reference taxon, and then compares the abundance of every taxa based on that. And then it would take the next one, say a Bacteroides genus, and it would take that as a reference taxon and compare the abundances, while the other microbes will using that as reference. And what this means is that it generates what's called the W statistic, which is the total number of times the taxon of interest was identified as significant during all of the tests that it ran for that taxon. And so this is important because the W statistic is then used to identify the proportion or the number of total tests it ran And it compares that to the W statistic to identify which taxa are significant. For example, it would say in 90% of the tests I ran, this taxa was significant. And I'll say yes or no. And there's 3 different cutoffs generally used for ANCOM, 90%, 80%, and 70%. And depending on how stringent you want your analysis to be, depends on which cutoff you want to use. But I would generally recommend using the 90 or 80%. However, the unfortunate truth to all of this is that no matter what tool you choose, you're always going to end up, in many cases, you're going to end up with a different number of significant features depending on the tool you chose. And so what I mean by this is in this heat map on the x-axis, we have the number of different differential abundance tools. And on the y-axis, we have a number of different data sets, 16S ribosomal RNA sequencing data sets. And the numbers in each of the cells represent the number of significant features that tool identified in that data set. So if we look at this first row here, the human C. diff data set, which compared healthy individuals to individuals C. difficile, if you look across that row, you would see there's a pretty highly variable number of microbes that are identified as being significantly different, depending on the tool that was chosen to test. And so this is important to take into account when doing this doing differential abundance analysis, as the choice of tool you make can have an impact on the biological conclusions that you come to when looking at the data sets. And so one solution to this is actually using a number of different differential abundance tools and reporting the results of each tool tested. So you might be interested in testing both ANCOM and ALDEX2 on your data sets, which are compositional, a bit more conservative in the number of microbes they identify as differentially abundant. And then you might also be interested in using a proportional based analysis like Maslin-2 and metagenome-seq to look at a different aspect of whether or not there's specific microbes that are different, that are significantly enriched in one group or another. Overall, I would say when comparing to previously published literature, you should always use the same differential abundance tool, or at least one of the tools you test should be the same as the one that was previously published, because as previously shown, if you don't, this can be a factor that results in different results than what has previously been shown. And finally, the most important thing when thinking about differential abundance or even any of these statistical tools in general is that you shouldn't blindly trust the statistical tool you're using. You shouldn't just assume that the tool is perfect and it's going to pull out the correct relationships. You should always visualize the data to help you understand the relationships that these statistical tools are indicating. This will give you a much better idea of what exactly is going on in your data. And there are a number of different visualization techniques. One that I commonly use is R, the programming language R. However, Qine2 also has a number of different visualization tools that can be used to inspect your microbiome data. In the second part of the lecture, I'm going to be doing a brief introduction to machine learning in the context of microbiome data. Before we get into the meat and bones, I'd first like to highlight maybe why you might be interested in machine learning using microbiome data. So in a study in 2019 published in Nature Medicine by Thomas et al., they compared individuals that either had colorectal cancer or were healthy individuals. And they're interested in using machine learning to see if they could classify whether someone had the colorectal cancer or not. So in this plot here, what we're looking at is what's called the receiver operator curve. And on the x-axis, we have the false positive rate. On the y-axis, we have the true positive rate. So the false positive rate is the number of times the classifier identifies as someone having cancer, but it is actually a healthy individual. And the true positive rate is how many times the classifier identifies correctly that the sample that it came from was from someone that had colorectal cancer. The two dots on this plot represent two tests that are currently clinically used for the early detection of colorectal cancer, one being the fecal blood occult test, and the other one being the WIF1 methylation test. And you can see that their true positive rates and false positive rates are actually surprisingly low considering that they're clinically used. However, what the lines represent is their machine learning models based on microbiome data. So in the green is a model based on microbiome data alone. And then in the yellow and purple or pink there are two different models based on the microbiome data plus the results from either the fecal blood occult test or the WIF1 methylation test. And so you can see based on the results of this paper that microbiome data actually does have the potential to increase our ability to accurately diagnose colorectal cancer compared to healthy individuals. And so how exactly was this done? Well, in general context, a machine learning workflow works as the following. You have some sort of data, in this case in microbiome, you might have the abundances of various ASVs or the taxonomic abundance of various microbes. You might have predicted pathway abundances from a tool such as PyCrest2, which you'll be talking about at length during one of Dr. Morgan Langel's lectures. And then, of course, you had the metadata of interest. So in the previous case, it would have been individuals with colorectal cancer versus individuals without. You then take this data and you split it between a training data set and a testing data set. In general, the training data set will comprise of the majority of your data, generally around 80% of it, and will be used for model training, whereas the testing data set will comprise of about 20%. And it's important to leave out a testing set as you need to use this testing set to validate the model performance. So how well your model might perform in the real world when it's tested on data that it's never seen before. So using the training data, you would train some sort of machine learning model. There are various machine learning models that exist and a number that are common in microbiome literature. Fortunately, there's a QIIME2 plugin called the QIIME2 sample classifier plugin, which actually implements a number of commonly used microbiome machine learning methods, including random forest models and I believe support vector machines. But basically how these models work is they take in some data, they then learn various patterns through that data to then use those patterns to identify whether a sample came from someone that has the disease or someone that doesn't. So once you've trained your model, what you're going to do is take your testing set and give it to the newly trained model to see how accurate your model is. So a number of different ways you can identify this is the accuracy. So how often your model correctly makes the decision whether or not someone has the disease or not. Or also, you can also compare them using receiver operator curves, which I highlighted in the previous slide, where those are the curves with the true positive and false positive on the different axes. And what I really want to emphasize here is that it's extremely important, this final step in testing your data. And that's because without using it, you can end up with models that are overfitted or have overinflated accuracy. Fortunately, in the QIIME2 sample classifier plugin, it already does this training and test data split for you. However, if you aren't using the QIIME2 sample classifier and doing this on your own using some sort of program or package in R, I highly suggest using a testing set, as it'll help you identify whether or not if you were to train a model and then go collect a sample from a new patient, how well it might perform on that sample from the new patient. And in some cases, you might even be able to do further validation or different types of validation. So if you have a smaller data set, sometimes you can use what's called leave and out validation. If you're interested in doing that, I suggest checking out Microbiome Helper. It's Microbiome Helper, which is our lab's set of tutorials for different machine learning, for different microbiome analysis types. And there's also Leave One Data Set Out analysis, which is very similar. And it's actually what the Thomas et al. Paper used, where you'd have a cohort of interest and you train all your data of all the samples in that cohort, and then you test your model on a different cohort that the trained model had never seen before. And with that, I'd like to thank everyone for listening. I look forward to working with all at CBW 2021, and I'd be happy to answer any questions on Slack. You can go ahead and direct message me if you'd like. My name on Slack is, of course, Jacob Nearing. Thanks, everyone, and bye.